Hi, I'm Julie Miller, and I'm going to tell you how I found that modularity and connectivity of nest structure scales with colony size. So today I want to talk to you about the consequences of evolving larger body size. Getting large can result in challenges in transporting resources and information, but life has mitigated these challenges through a variety of structural solutions. For example, multicellularity has allowed organisms to overcome constraints on the size of individual cells. And once multicellular systems became large, they then needed specialized vascular networks to deliver resources across greater distances. And other multicellular systems have evolved larger size by innovating body plans to increase their surface area. And we can extend this analogy to the groups of social animals who may face similar challenges of transporting information and resources across greater distances. And the context of this movement is the nest, which may also shape interactions and the efficiency of transport. So here we examine these dynamics in ant colonies. And there is already good evidence that the structure of ant nests in particular shapes the behavior of the colony. For example, in foraging and recruitment and in nest evacuation during an alarm event. So the question I tackled here was, how does nest architecture change with group size to maintain transport in nests of larger colonies? And I proposed three mechanisms for how nests might accomplish this. So the first, subdivision hypothesis, proposes that nest chambers physically compartmentalize individuals into smaller subgroups so that these subgroups are small enough that individuals maintain rapid contact and there isn't this viscosity of having to move through large crowds. And this hypothesis predicts that as colony size increases, the number of chambers in their nests ought to increase, but that the size of those chambers should not. And so to test this, we comb through the literature of looking for nest castings, images, excavations, focusing only on the ground nesting ants. And we first counted the number of chambers in each nest, and then we measured the width of all chambers in each nest, but we standardize these measurements with the size of the ant of the species that resided in those nests. And then we compared these two measures with colony size while controlling for phylogenetic relationships. And what we find, as predicted, the number of chambers does indeed increase with colony size, but, the size of those chambers remains constant. And this satisfies both predictions of the subdivision hypothesis. So the next two hypotheses relate to the topology of the nest, or how the tunnels are connected. And they are both ways in which the nest could reduce travel distances. And so I'll explain how we tested both of these hypotheses together. So first, in order to quantify the structural features of nests, we first had to convert nest images and castings into networks. And so we did this by assigning each chamber a node and each tunnel as an edge. And so to consider both of these hypotheses, the branching and shortcuts hypothesis, we wanted to understand how the connections in a nest affect how easy it is to move from one part of the nest to another. Therefore, we focused on this measure, the mean distance of a network. And just to define this technically, the mean distance is the sum of the shortest path lengths between all possible pairs of nodes. And then we divide that sum by the number of all possible node pairs. And so just to illustrate in an example here, the distance between these two nodes is 1, and the distance between these distant nodes is 12. They have to pass through 12 edges to get there. And we repeat this process for all possible combinations. And as you can see, the size of the network will affect the mean distance. So these larger networks have a larger mean distance than the smaller ones. But for networks of the same size, these networks here each contain 10 nodes. Adding a branching structure will reduce the mean distance. But you can also reduce the mean distance further by adding a shortcut, or what we call a cycle. And by adding either branches or cycles, it becomes easier to access more distal parts of the nest. And that's what this mean distance is attempting to capture. So what I've shown you here is that both size and structure affect the mean distance. And we wanted to be able to control for both the effects of size and structure as we set expectations about how mean distance might change with nest size. So to do this, we created 3 reference models based on different rules for how networks grow. The first is this chain network, which is really represents the lower end of connectivity. Everything's just in a line. The triangulated reference models represent this upper end of connectivity, and they're highly connected. They create these cycles or these loops. The entire network consists of a bunch of triangles. The minimum spanning tree networks are sort of an intermediate level of connectivity, and they consist of a bunch of branches but never contain any cycles or loops. So we generated expected values of mean distance for each reference model for the networks between 2 and 100 nodes. And then we plotted mean distance values for our observed nests, and we see that they sit somewhere in between the chain and the minimum spanning tree networks. And in order to get back to our original goal of understanding how these structural features change with colony size, we needed to scale our observed nests by each reference model. And we did this by dividing the observed mean distance of our nest by the mean distance of the reference model of the same size. And we compared those standardized values to colony size here in this figure. And so let me walk you through this figure because there's a lot going on. I want to first call your attention to this gray dotted line at 1, the horizontal dotted line. And values that fall on this line indicate when the observed nest and the reference model have the same mean distance. Values less than 1 indicate the observed nest had smaller mean distances than expected by the reference model of a given size, and values greater than 1 indicate that the observed nest had larger mean distances. And so that's to orient you. Now, I want you to focus on this red line, the data in red. And this is data that's been normalized to chain networks. And what this tells us is that in small colony species over here, mean distances matched or very similar to the values expected for chain networks. And indeed, most of those nests are chains. But in larger colony species, as we move down this x-axis, mean travel distances became shorter than would be expected for chain networks of those size. So these findings indicate that as colonies increase in size, their nests become more branched than the chain equivalent. And so there's a big but there. These shorter mean distances could also be explained by additional shortcuts, additional cycles. And so that brings us to our third hypothesis that proposes that large colony species have more shortcuts in their nests. And what we find when we count the number of cycles in a nest is there are not significantly more cycles in the nests of larger colony species. But like before, we wanted to standardize these results with expectations from a reference model for how the number of cycles should increase with nest size. So we standardized these data to a triangulated network. And what we find, again, is that nests from larger colonies do not contain proportionally more cycles. So to summarize, I've shown you that the nests of larger colony species are more subdivided, and they reduce travel distances by increasing branching, but not by adding shortcuts or cycles. And these results highlight the similarity in how ant nests and the bodies of unitary organisms respond to increasing size. And if you'd like to read more about the details of this work, you can check out the forthcoming publication in Evolution. And with that, I'd like to thank first all of the people doing this really important natural history work that made this work possible, and also my lab mates, and especially Emma Wan and Sean O'Fallon, co-authors who collected a lot of this data. And with that, I'm happy to take questions in person.